For joining us. If this is your first time, welcome to the Conservation Science Seminar. This is a weekly seminar that the Conservation Science Program puts on, where we invite a variety of different speakers on a variety of different conservation-related topics. This week, we are excited to have Dr. Dan Larkin from the Fisheries, Wildlife, and Conservation Department. He is an associate professor, and he and his team conduct applied research in invasion biology and restoration ecology in lakes, wetlands, woodlands, and prairies. Through his extension program, he trains volunteers and professionals to support early detection and management of invasive species and ecological restoration. And today the talk of his title is The Thin Line Between Native and Invasive in Aquatic Plants, Implications for Surveillance, Outreach, and Management. So with that, I'd like to formally welcome Dr. Dan Larkin and go ahead and take it away.
Okay, great. Thanks, Wes. So I'm going to cover a fair amount of ground today, and it's some stuff I've been sort of stewing on for probably about the past 10 years and think a lot about in terms of my research and extension programs. And here's an overview of what I'm going to cover today. I'll talk about the ecological consequences of macrophyte invasions, the prevalence of there being close native relatives and hybridization in macrophyte invasions, talk about a couple of invasive species as case studies, and then expand to sort of a broader hypothesis for why I think there are what I describe as thin lines between natives and invasives in aquatic plant invasions. And then I'll describe a test of this idea and close with implications and needs for extension and applied research. So first off, what's a macrophyte? Aquatic plants and macroalgae. So these can span from plants like cattails or other emergent plants that occupy moist soil and wetlands out into floating and submerged plants that grow in open water in lakes. So that's the focus for today. And invasive macrophytes, why should we care about them? They're increasingly prevalent. So this is data from the Minnesota DNR showing the number of water bodies that have 4 priority and aquatic invasive plants. And notice that this is on a log scale. So this is actually showing some pretty rapid accumulation of new populations. And this is also really an underestimate because there are other species that are much more prevalent and would sort of be off the scale, but are not tracked so closely. So our lakes are increasingly invaded, and that has a number of important consequences. It can interfere with the way people use lakes. It has economic impacts in terms of control costs, property values. It can reduce habitat quality for fish and wildlife. And what motivates me personally is threats to native plant diversity. As a plant ecologist, this is where my interests are concentrated. So I'll talk a little bit more about those ecological consequences. So we've done some work on looking at plant community impacts of invasive macrophytes, and I'm going to cover a couple of papers that were led by Ranjan Muthukrishnan, who many of you knew when he was a postdoc here, and he's now at Indiana University. And we looked at some statewide trends in invasions and native plant diversity, leveraging data from the Minnesota DNR. So we had vegetation surveys from over 1,100 lakes sampled over a 13-year period. And this is what the distribution of those records looks like, with both invaded and uninvaded lakes distributed throughout the state. And then importantly, some lakes for which we had not just a single survey, but had multiple years of data, so we could look at temporal patterns. So here's some of what we found. One, that the most diverse lakes were most vulnerable. That is, lakes with greater native richness were associated with greater invader richness. And this is a pattern that's been documented in a number of terrestrial habitats. And it's understandable because really, environmental factors that are making a habitat conducive to native species are also, in many cases, going to make it conducive to non-native species. But it means that those lakes that have the highest plant biodiversity value are also going to be invaded most. So that's at a large spatial scale, at a lake scale. What about finer scale interactions? So looking at points within lakes, which is sort of the neighborhood scale at which plant species are interacting with each other. What we would like to see is native diversity conferring some resistance to invasion, which is called biotic resistance. But we do not see that pattern. The hope would be, as you have higher native species richness, you would have lower probability of invasion, but that is not what we found. Conversely, the arrival of an invasive species, that when you compare pre-invasion to post-invasion, did reduce native diversity. So we did see evidence of competitive displacement or competitive exclusion by invaders. So this is sort of a lose-lose proposition for native plants, where they're not really resisting the invasion, but then can be displaced by the invasion. And I should say, huge amount of noise in the data, but nonetheless, this significant negative relationship between invasion and native richness. So that's what's going on in terms of the numbers of species, but there are additional changes to lake plant diversity that are important. And in particular, we found that lake plant communities are undergoing biotic homogenization. So what do I mean by that? So I'm going to use this sort of cartoon forest. We have a landscape. We have 4 different forests. Each of those forests has 4 tree species represented with the different colors. And that's their alpha diversity, their local diversity. We also can compare the composition of the forest and see that they're compositionally distinct from each other. They share some species, but not all. Each forest has its own composition. In contrast, we could have a different landscape where we have the same species richness, the same alpha diversity, but the forests are not distinct. And that would represent a loss of beta diversity. And biotic homogenization is, over time, different ecosystems losing their distinctiveness, or different communities losing their distinctiveness and becoming more similar to each other over time. So degradation of diversity. So in this plot, I'm going to show these changes in similarity over time using the temporal data. And this is a measure of similarity in community composition. Where there are more species in common, you get higher values. That represents lower beta diversity. Fewer species in common would be lower values. So first off, lakes in general became more similar to each other over time. And this is showing the change for uninvaded lakes. And then this is really ramped up in invaded lakes, which are less distinctive and show a little bit more homogenization over time. Beyond, sort of caring about what's happening to the native plant communities, these changes in both alpha and beta diversity can have negative effects on ecosystem resilience and functioning. Okay, so that was to sort of motivate you to care about macrophyte invasions. And now I'm going to talk about this phenomenon of there being close native relatives and hybridization in invasions. And like many things, we can go back to Charles Darwin, who described sort of the evolutionary legacy that can be imprinted on invasions. And this idea of the pre-adaptation hypothesis. So that's that non-natives succeed where closely related natives are present because they share traits that make them pre-adapted to the new environment. So in other words, if I have close relatives who are already thriving in a habitat, I'm likely to share some ecological similarities with those close relatives, and therefore that might be good habitat for me. Can leap forward about 140 years before a paper that delved into more specific examples from the system that I'm talking about today, and that's our colleague Sue Galadowicz and others here at the University of Minnesota published this really influential paper on 5 different wetland invasives in North America. And they describe the importance of formerly allopatric populations becoming sympatric in disturbed landscapes. That is, going from being geographically separated to geographically co-occurring, and described that there was hybridization and introgression happening in these invasions that was leading to phenotypes or characteristics of plants with selective advantages. Hybridization meaning crosses of different genotypes within species or across species, and introgression being back crosses then from those hybrids to parental species. And this idea has been really well supported over the past 20 plus years. And in fact, I first gave a shorter version of this talk at a wetland symposium a couple of years ago that was a 20-year retrospective of this paper. So for example, purple loosestrife, there's evidence of possible hybridization and crossings of multiple introductions driving the invasiveness of this species. Typhax glauca, or hybrid cattail. This is a hybrid species that's the hybrid offspring of a native cattail and a non-native cattail. And then introgression is widespread, that is back crosses to the parent species. And you end up with what's been described as this hybrid swarm of mixed native and non-native genotypes. And hybrid cattail is a highly invasive species. Reed canary grass is a species that is native to North America, and also there are non-native genotypes that have spread here, and the invasion in North America has been associated with genes of European origin. But I also want to point out that there's work going on here in horticulture that is sort of examining this and finding some different patterns with respect to origins of invasive populations. And then Phragmites australis, common reed, another species with a really wide native distribution, a circumglobal distribution, native to every continent except Antarctica. And these different genotypes have arisen in these different locations and then been mixed around by human activity. And Phragmites is really the poster child for this term I'll use of cryptic invasion. That is an invasion that's sort of happening beneath our eyes that we're not picking up on because it's hard to detect. Because in this case, it's already a native species here in North America, but there's been this spread of non-native genotypes of European origin that require genetic testing to identify. So Kristen Saltonstall published a really influential paper in 2002, and she used contemporary sampling of populations as well as herbarium records to show this pattern of spread of an invasive genotype. So you can see that on the right, there's just a little bit of the non-native Phragmites concentrated on the Northeast Atlantic coast before 1910. But over time, that really spread inward, westward, and expanded. And now there's high distribution throughout the lower 48 and into Canada. So this is something that Sue and I and Julia Bonin have been working on with MACERC support over the last few years. And one of the things we wanted to do is determine the distribution of this non-native Phragmites in Minnesota to support statewide control efforts. So we did crowdsourcing, had people send us in samples from around the state, and then our own targeted search effort. And here's what we ended up with. This is the distribution of non-native Phragmites as we currently know it today in Minnesota, with the different colors being whether or not a population has been treated yet. So we're working with the DNR to try to deploy an ambitious control effort, because there is a lot of the state that really doesn't have invasive Phragmites yet, but we'll get it over time. But Minnesota is also a state that's really rich in native Phragmites. It's an important native wetland plant that's widespread throughout the state. So there's clearly, you can see there's a lot of co-occurrence of these genotypes. And that leads to the question of, is there hybridization between native and non-native Phragmites? You would think there would be, but it's been found that native and non-native have actually remained pretty genetically distinct in North America. However, they can be crossed in the greenhouse, so we know it's biologically possible, but there have been few examples found in the wild, although more are popping up. And one thing that's been suggested is maybe we're not looking in the right places or using the right molecular markers. So a lot of the search for hybrids has been in places where native Phragmites is largely already extirpated. And the Great Lakes in particular were highlighted, Great Lakes region highlighted as an area where more of this exploration should be done, since we still have a lot of native Phragmites. In addition, maybe we've been using molecular markers that are too coarse. And since this paper was published in 2012 about this issue of hybridization in Phragmites, a lot of new resources and tools have emerged to enable that. So we partnered with the group in horticulture doing the reed canary grass research to do some genotyping by sequencing to get some higher resolution genetic data for Phragmites populations in Minnesota. And here, this is showing the populations that we tested through this genotyping approach, native in orange, invasive in blue, and then about a dozen cases where we have possible hybrids, about 4% of samples. And so this lack of a clean separation between native and non-native is potentially a complicating factor when we have this breakdown between the subspecies. In addition, research in the Western US has suggested there can be hybrid vigor in dispersal, establishment, and growth. For example, here showing that hybrid Phragmites produced a lot more seeds. So this just sort of adds to the urgency of being proactive in control efforts so that there can't be sort of more of this hybridization potentially leading to this hybrid vigor. Another species where hybridization is important is in Myriaphalum, water milfoil, and specifically Eurasian water milfoil. And it was shown that Myriaphalum spicatum, Eurasian water milfoil, can hybridize with native Myriaphalum species. And in particular, what we're concerned about around here is crossing of Eurasian water milfoil with native northern water milfoil. This is, again, a highly cryptic invasion. You end up with this intermediate morphology where plants look something in between the native and the non-native. It requires genetic testing to confirm. And the situation is even more challenging than with Phragmites because it's a submersed species. So you can't see it when you're driving by on the highway. You have to really go out looking for it. And this hybridization is really consequential for management. This is work from Ryan Thum and others. Ryan is at Montana State and has done a lot of work on water milfoil genetics and has documented this issue of herbicide tolerance. So this is showing a lake in Michigan before and after an herbicide treatment. And you have the distribution of Eurasian water milfoil on the left, and then you can see the treatment was incredibly effective. It knocked out the Eurasian water milfoil. Unfortunately, this lake also had hybrid water milfoil, and there was not effectiveness for hybrid water milfoil. So it's become a real significant issue in aquatic plant management in the upper Midwest, this herbicide tolerance. We were interested in whether there, beyond this herbicide tolerance, there might also be increased invasiveness. Is there hybrid vigor in its invasiveness? And that had been suggested by small-scale laboratory trials and anecdotal reports, but it hadn't been investigated within lakes. So we evaluated this by comparing the phenology and performance of the hybrid and parent species in 9 Minnesota lakes. And when I say we, I really mean Wes Glisson, who's a researcher in my lab. So what Wes found is evidence consistent with there being hybrid vigor and greater invasiveness. So for one, higher surface matting in hybrid than either parent species. So the hybrid in the middle, reaching the surface, branching laterally, and causing more matting on the surface, which is one of the things that interferes with recreation and then can also have consequences for native vegetation that gets shaded out and overtopped. In addition, we found higher potential for sexual reproduction with much greater production of flowers in hybrid water milfoil than either parent. So in this case, we see these challenges. Detectability is lower because of cryptic morphology. Control is less effective in this case because of these herbicide tolerant strains. The impacts are potentially worse because of hybrid vigor. And then also a theme I'll return to later in the talk is that the messaging is more complex. So I've been thinking about this over the years, and I think beyond these specific 5 species from Sue's paper that I've highlighted, I think it's a more general phenomenon in aquatic plant invasions that spans from the saturated soil of wetlands to the open water of lakes. And from these sort of thin lines, as I'll describe them, where you have close relatedness. So for example, Potamogeton crispus, curly leaf pondweed, is one of our lake invaders. To our knowledge, it doesn't hybridize with native Potamogeton species, but it invaded a region that's already home to 31 native congeners, that is, other species in the same genus. So clearly this is, sort of Potamogeton country where this genus does well, suggesting there might be this phenomenon of pre-adaptation. And then beyond these thin lines, you can have a breakdown where there is genetic exchange, where you have sympatry and you have hybridization, as work by Jasmine Eldaweli and Ray Newman and Ryan Thume has shown, with this concentration of lakes having these different water milfoils, and even within lake, having the hybrid and parent species co-occur. Okay, so why might this be happening? And is it the case, as I'm contending, that it's happening more broadly across aquatic plants? So Luis Santamaria, in a really influential paper, described the fact that aquatic environments are stressful for plants. It's hard to get carbon, there's sediment anoxia, there's light attenuation in the water column, and that this has selected for broadly tolerant plants. In addition, we have these environments that are heterogeneous at fine scales, but often similar across large scales. So within a lake, along a depth gradient, the environmental conditions for plants can really vary. But across large regions from lake to lake, there can be a lot of similarity in environmental conditions in these relatively closed systems. And so he posited that this had led to a global aquatic flora with broad tolerances, wide distributions, and limited taxonomic differentiation. So in other words, it might matter less that a plant is trying to grow in a lake in Minnesota versus a lake in Sweden than just simply that a plant is trying to grow in a lake at all. So to expand on this, I've made this little cartoon. So let's say we have some pool of plant species, each represented by a different circle, and we're interested in colonization from this species pool into specific habitats, a process called community assembly. We can imagine that maybe a lot of plant species can colonize into terrestrial habitats, that the filter that takes out species that can't live on land might be relatively weak, and only some species will be rejected. In contrast with colonization of an aquatic environment, that filter could be much stronger and fewer species get through it. Now, obviously, there are many more environmental factors that are important, these other axes like light availability and nutrients, biotic interactions within a habitat. But I would argue that those are potentially secondary limits or secondary filters compared to this first overarching filter of can you live under these wet conditions. If this is the case, it would suggest a stronger influence of pre-adaptation in aquatic invasions than in terrestrial invasions. And coming out of that, we'd expect to see a pattern of aquatic species being an evolutionarily distinct subset of the flora that have this ability, and aquatic invasives being more closely related to natives than terrestrial invasives are. So I was interested in exploring those ideas, and so I tested this by comparing the relatedness of native and invasive species in terrestrial versus aquatic plants. And I used Minnesota and Wisconsin as the case to explore, share a lot in terms of biomes, and are both, really environments that are very rich in aquatic and terrestrial habitats. I identified priority invasive species, that is, species that are both non-native and considered harmful that have been found in the region growing in the wild, so can establish here. And I used regional watch lists, including from a couple of our centers on campus and other sources, and a paper that I wrote previously, and identified 126 terrestrial invasive species to focus on and 27 aquatic species to focus on. I then downloaded all the herbarium records for all of the seed plants that have been collected in Minnesota and Wisconsin. So kind of the complete flora of those states, which includes these focal invasives that have been found here and for which samples have been submitted to herbaria. And then I used herbarium records and a published mega tree, a giant phylogeny of the seed plants globally, to build a phylogeny for these two states, for all native and non-native upper Midwest seed plants. And that was about 5,000 species. Of those, about 4,000 are terrestrial species, and about 1,000 I classified as aquatic species based on wetland indicator status, which is this classification that's used in wetland delineation and other applications to identify species that are dependent on hydric conditions, hydric soils or aquatic conditions. And so before I dive into the tree that I built, I just want to describe a little bit more about what a phylogeny is and how to read one. And I think of it as essentially a molecular family tree. And I should say at this point, I'm a plant ecologist, not an evolutionary biologist. So with that caveat in mind, I'll proceed. And I thought the waterfowl would be a relatable example for a lot of people. So here is a phylogeny of waterfowl by Lee and Clark. And some characteristics of a tree like this, you have these nodes. Here I've highlighted one. That's the common ancestor for all the subtending tips, all the species that are downstream of that node. And as a group together, those species with a shared common ancestor represent a clade, in this case, the true geese. Or we could go back in time a little bit evolutionarily and broaden to a broader clade by adding the swans, a sister group, and then we have the Anserini, if I'm pronouncing that correctly. What's important is that closer relatives are separated by shorter branches. They have shorter time or evolutionary distance between them. So for example, mallards and black ducks are very close relatives in this tree. And that distance between each species has a closest relative. In the case of mallard and black duck, it's each other, and that represents the nearest taxon distance. And I'm highlighting that because that's a metric I'll use in my analysis. And then another important concept is phylogenetic niche conservatism. That is the idea that closer relatives will be more ecologically similar to each other. So for example, the mergansers and allies are an evolutionary group, and they also have this diving, feeding behavior that makes them ecologically similar in terms of their niche they're occupying. And I'm just throwing on some asterisks there to acknowledge that there are many exceptions and many important ways in which close relatives diverge. But as a general rule of thumb, as you look across larger taxonomic scales, across more species and across more traits, you'll have this general pattern of closer relatives being more ecologically similar. Okay, so this is the tree that I wound up with for the upper Midwest seed plants. And here are a few of the major lineages within the seed plants that are represented here. And then here I've color coded the species to indicate whether they're aquatic or terrestrial. And just a couple examples of a terrestrial invasive, Lonicera tartarica, and a aquatic invasive, where these live on the tree. So then with this tree built, I asked a few questions. One, are aquatics phylogenetically distinct? And for that, I tested for what's called phylogenetic signal, that there's sort of non-random sharing of traits based on relatedness. And I also explored whether aquatic invasives are more closely related to natives than terrestrial invasives are. And I did two tests, a taxonomic test, looking just at the proportions of invasive species with native congeners, that is other species in their same genus, and the proportions with native conspecifics or hybridization partners. That is cases where we have native and non-native subspecies of the same species, or different species, but closely related enough that they can produce viable hybrid offspring. And then as paired with this taxonomic test was a phylogenetic test where rather than this categorical counts, using phylogenetic distance to look at relatedness. So first, are aquatics phylogenetically distinct? So here we have just the aquatics highlighted with these blue lines, and we're basically asking sort of a spatial question. Are aquatic species more closely related to each other, clustered within certain lineages than you would expect by chance? And the answer is yes, they're a clustered subset of the total flora. Are aquatic invasives more closely related to natives? So using the taxonomic approach, I found that relative to terrestrial invasives, aquatic invasives had higher frequencies of native congeners, close to twice as frequent, and then many more cases of native conspecifics or hybrids. Same question using the phylogenetic approach. Here we have aquatic and terrestrial subtrees with the invasives denoted with these red symbols. Each invasive has a closest native relative, nearest taxon distance. And I compared the observed values for each invasive to a distribution of nearest taxon distances for the native species pools, using essentially a z-score or a standardized effect size to see how closely related are they relative to expectations for these species pools. On this figure, species that when on average we have species being more closely related than expected by chance, we'll get negative values. If less closely related than expected by chance, we'll get positive values. And for terrestrial invasives, we found that they are more closely related than expected by chance, but that pattern was much stronger in the case of aquatics. They were significantly more related to each other, to native relatives, than terrestrial invasives were. Okay, so now I'm going to kind of turn to what I think some of the implications are of these basic biological patterns for invasive species response efforts and for research and extension needs. So I think the evolutionary legacy of adaptation to aquatic environments poses some special challenges. One, outreach and communication is challenged by these finer distinctions between native and invasive. So there's a lot of nuance to communicating that you have this distinct lineage or genotype that's problematic versus a species being, wholly novel. So in the case of silver carp, you have these fish jumping out of the water, hitting boaters on the head, doing something that it hasn't been seen before ecologically. And for that, I think, kind of the classic, quote unquote, the aliens are coming type of invasion biology rhetoric is, sort of makes sense. You can buy it. But when you're talking about these genetic changes that can be consequential and important, but are more cryptic and subtler, that's really not a fit. And you can't avoid sort of the gray areas. Another thing I see is this issue, what I'll call type 2 error, borrowing from statistics when you fail to recognize a significant distinction. In this case, lumping native and non-native, or using a visual metaphor, painting with too broad of a brush. And this can cut both ways, and I see this come up around Phragmites. So one type of type 2 error is not discriminating against the invasive. So this paper, haplotype M, that's a European genotype, and these authors say it's the most common variety worldwide, and it is the ancestral source of North American varieties. All common reed varieties provide critical ecosystem services. So this is essentially saying, let's think of this as a species, look at it at the species level and say, this species is a good thing, not recognizing the important ecological differences that are present between native and non-native Phragmites. But it can also go the other way. We had a study where we provided genetic testing services for managers around the country, and we also surveyed them on their management plans. And we found that 11% of native Phragmites stands were being treated with herbicide or were planned for treatment because managers hadn't sort of taken the care to determine whether or not they actually had the invasive variety, which is really unfortunate. Another implication is surveillance is difficult. Surveillance is challenged by these cryptic invasions that can easily escape notice or require expert verification or tools. This paper in 2018 reviewed this phenomenon of cryptic invasions in the literature, showed growing interest, and also showed that about 80% of the examples were from aquatic ecosystems. So this is a particularly notable issue in wet environments. For control is challenged by the risk of non-target impacts to close native relatives. So in aquatic plant management, the goal is specificity to be able to kill the target species without causing collateral damage to native species. That becomes more difficult if you have very close relatives present that are likely to be growing in the same places at the same times, having susceptibility to the same active ingredients. Conversely, failure to control can lead to native relatives being displaced by competition or this genetic introgression. Going back to the Salt and Salt paper, this is zooming in on southern New England and showing the sequence of native Phragmites being displaced from that landscape with the spread of European genotypes. So there are a lot of applied research needs that come out of it that we've been working to address. One is simply the question, how bad is it really? An example of a species that we've been working on a lot over the past few years is Starry Stonewort. This was not in my phylogeny because it's not a seed plant, it's a Macrophyte, a type of macroalgae, but it's also a cryptic invader. Because there are a lot of native Charophytes, it's difficult to identify, they can be difficult to identify as species. And so it was first found in Minnesota in 2015, but it may very well have been here earlier and just sort of escaped people's notice because they lumped it in as just some sort of Chara species. When Starry Stonewort has been found in new states, it often leads to a great deal of concern and alarm. So this is a headline from Wisconsin where it was found the year before. An invasive species that spreads fast. It's the start of the death of a lake. But at the same time, Starry Stonewort, you learn it's rare, even in an endangered species in its native range. It's conservation priority in many countries. It's considered an indicator of good water quality that makes important ecosystem and food web contributions. And so we had these sort of strong qualitative cases on both sides that Starry Stonewort's very bad or that we shouldn't be worried about it. But there was a lack of actual rigorously collected quantitative information on its impacts. And so Carly Wagner focused on this for her master's, and some of you saw her defense a couple of weeks ago. And she decided to take on this question of what are the plant community impacts of Starry Stonewort? And what she found is that indeed native macrophyte abundance and diversity do decrease with increasing Starry Stonewort abundance, but also that there's some there's variability from lake to lake and year to year, and there's some potential for recovery of native species. So not surprisingly, sort of in between those extremes of it's the death of a lake and it's nothing to worry about. We also need more applied research on control efforts. Are they working? How can we improve outcomes? Many of you know Mike Verhoeven, doctoral candidate. For his master's, he looked at evaluating the effectiveness of herbicidal management of curly leaf pondweed, something we've been working on controlling in Minnesota for decades, to investigate sort of what are the constraints on effectiveness. And what he found is a lot of the variation in outcomes depended on things that were beyond managers' control. So for example, overwinter conditions, sort of how severe or mild a winter was, has a lot of effect on curly leaf growth. Lake trophic status, the productivity of a lake, has a lot of effect. But then there is variation that managers can control, and differences based on management actions. So this figure is showing the lake-wide extent of curly leaf pondweed with consecutive years of treatment on the x-axis. And Mike found that control requires a sort of sustained multi-year effort to bring down the lake-wide extent. Another important question is how much does control help versus hurt native species? Is the cure worse than the disease, for example? So he'll have some forthcoming work on that. We also really need more early detection. We need to be able to find these nascent populations at times when control is more possible. And I'll return to Starry Stonewort. So this is some work in progress by Wes again. And this is showing invasion trajectories in 3 states, Minnesota, Wisconsin, and Indiana over the past 10 years. Each of these lines represents one of 35 lakes in this data set. And Wes is synthesizing and examining this data to look at patterns in terms of management outcomes. You can see just from this figure that we're seeing really rapid expansion of this species within invaded lakes. But unfortunately, once Starry Stonewort becomes widespread in a lake, control efforts are not reducing its extent in general. So here we're seeing the extent before and after treatment. We would like that second bar to be substantially lower, but it's not. So most of our control effort is with copper-based algicide. It can provide local relief through intensive effort, but it is not scaling back that expansion in the way that we see with curly leaf pondweed, for example. But we know that we can contain populations if they're found when they're small. So this is a lake where Starry Stonewort was found in 2017 in a program I'll describe shortly. And the Minnesota DNR has been going out and performing hand removal of this Starry Stonewort. And this figure is showing the amount of biomass they can find and remove over the last few years. And while it has not been eradicated, it has been contained. It hasn't been found elsewhere in the lake, and they're getting down to chasing these very small bits of it. So this really lies at the heart of a lot of our work in aquatic invasive species extension with my colleagues Megan Weber and Pat Mulcahy, where we're trying to increase this capacity for early detection. One of our programs is Starry Trek. We do this statewide single-day volunteer search effort. We have 20+ meetups around the state where local coordinators, who are usually AIS professionals or other really engaged folks, train and manage volunteers. And we do this in partnership with the Minnesota DNR and also University of Wisconsin Extension, which has a sister program on the same day each year. There are about 12,000 lakes in Minnesota, so obviously we can't search all of them. So one of the things we really focus on is prioritization for our limited search capacity. So another thing Ranjan did when he was a postdoc with me is develop this water chemistry-based risk model. And we combine that with input from our local coordinators who are, sort of experts on the ground and identify locations that they think are a priority for search. And we're finding that we're getting really good engagement, and people really seem to enjoy participating in this program, come back each year, and we're able to search more and more locations. And even with a pandemic year, we're able to carry it out by adopting some COVID safety protocols. And since we started this program, it's really been driving the discoveries of Starry Stonewort in the state. So we have about 15 known populations, and 4 of those have been found in recent years through volunteers conducting this simple search effort, including Grand Lake in our first year, that case where I showed the hand pulling. Starry Trek is an outgrowth of our core program, AIS Detectors, which was launched in 2017 and will soon cross into over 300 volunteers and professionals who have been trained and certified in kind of aquatic invasive species science and identification and reporting. Another outgrowth of AIS Detectors is we recognize this real need for people to become sort of more informed, savvier consumers of aquatic plant and AIS management. So this is a highly decentralized process in Minnesota and a lot of other places where it's driven at a very local scale by lake associations and their knowledge and capacity. And it's ecologically significant because we have a lot of lakeshore homeowners who are engaged in aquatic plant management, far less than half of whom have sort of lake-related professional experience. So I always think of this as kind of an ecosystem management version of the problem that I have if I go to Jiffy Lube to take my car for an oil change. And I know nothing about cars, right? So I just want an oil change. And then they're throwing all these other things at me, and $75 for this, $60 for that. And I'm not a savvy consumer. Well, this is the same situation, but much more consequential when you're making these resource management decisions. And so we're trying to provide a foundation for these folks to understand some of the basics around aquatic plant management, pesticides, how they work, how they're regulated, ways in which management effectiveness can be evaluated, and ways to sort of set realistic long-term goals. So many people, I'm sure more than are listed here, have been involved in various facets of these projects, and I thank them for their collaboration and as well as funding sources. And I will now stop talking at you and answer questions. I'm going to stop sharing so I can see people better in case anybody wants to turn on their camera and talk.
Thanks so much, Dan. That was a super interesting talk. I'll start off with a question from Sue in the chat. She says, it looks like Phragmites hybrids were located at the periphery of the Minnesota range rather than closer to the locations of the original introductions, where there would have been the greatest opportunity for hybridization. What do you make of that?
Yeah, it's a great question. I was really surprised when I first plotted that map of where they are, because I thought I would see concentrations in the metro and other areas where there's a lot of invasive Phragmites. One thing, so frankly, I don't know what to make of that. And I think it's something that we need to dig more into. Because I am surprised by that pattern. But it's a grass, it's wind pollinated, wind dispersed. And the map shows only those locations that we did genotyping of. And so it's somewhat misleading in that there's a lot richer background of overlapping native and non-native in general in this state that wasn't part of our genotyping.
Next question comes from Angelique. In the context of type 2 error, she's thinking about the hybrids and the idea of cryptic invasions in a game theory framework. And she's wondering, how bad are the consequences of misidentifying a population as native, invasive, or hybrid? How often do you think that arises, and what are the consequences for the future of the species?
Well, that's a difficult question. So I guess part of it depends on what the management options are and how effective those options are. So, for a species so let's say we miss identifying an invasive that we want to control, we also have to account for the fact that we may or may not be able to control that invasive very effectively. So the costs of missing an invasive we want to get rid of depend on our ability to get rid of the invasive. And then I guess on the other side of things, the kind of collateral damage side of things, would be hugely dependent on kind of the state of the species as a whole. Is it a strong, resilient population where we have a lot of individuals and they can bounce back from that unintended harm, or is it happening to a species that is maybe a more conservation concern and where we have, where we're sort of hitting them at the heart of their habitat. But I think both are unfortunate. One is sort of a negative consequence of being overly aggressive and kind of having too much of like a quick draw mentality for management. And then one is, sort of the flip side of that, the negative consequences of maybe being too passive.
Thanks, Dan. I was just curious kind of for your thoughts on that. Appreciate it.
Thanks.
Ray asks, should we be rethinking the plants as invasive genotypes rather than species? We do not see the non-invasive exotic genotypes. They do not establish.
Right, we do not see the non-invasive exotic genotypes, right. They're not the successful invasives. Yeah, I think, so that's sort of what led me to a lot of this thinking and to kind of want to explore this area, because that's how, to some extent, how I've started thinking about some of these invasive macrophytes, is that they're sort of in this messy gray area where they're on the edge of native and non-native. But that's also like sort of the exact opposite of what they tell us these days about how to communicate science, right? Is we're supposed to have kind of clear, understandable stories. And it's sort of the worst case of an ecologist thinking everything is a context dependent gray area. So I think biologically, I think there is a lot of value in thinking of it, not just at the species level, but thinking more in terms of genotypes and breakdown between species. But then I'm not sure, practically speaking, from an outreach and communication perspective, when you have people sort of limited attention, if that's an effective means of education and communication. And it can be taken too far, and I think it can, I think there really are important differences between native and non-native species, whether or not a species has been taken out of its historic range where it's evolved in. And I don't want to lose that by just saying, oh, we need to judge species by their genotypes, not by their origins. So I don't know, it's tricky.
Thanks, Dan. Megan asks, when comparing the phylogenetic closeness of terrestrial versus aquatic species, how did you deal with wetland plants such as Phragmites and purple loosestrife that tend to sit on the edges of those habitats?
Yeah, I treated them as aquatics. So basically for wetland indicator status, there's these letter codes, but it can be sort of facultative wetland, and then meaning the preponderance is going to be within wetlands all the way to obligate. And all of our truly aquatic and submerged species would be an obligate. So if it was facultative wetland and beyond, I treated it as aquatic. And I didn't attempt to break down within those finer categories because I think it was 26 or 27 of those species. So we just sort of run out of power subdividing them.
follow-up question. Did you have to go through hand code? Did you have to go through and hand code every single species in the analysis?
Yes, Megan, that's why I missed some of our internal deadlines. No, I'm kidding. I didn't. Luckily, there are a lot of really nice tools available through R for doing this sort of management of biodiversity data, as well as some websites that allow to reconcile species. So I was able to automate a lot of it, but that's where a lot of the grunt work was, in lining up, invasive species lists with what's in this published tree with what's in herbarium records and getting those to match.
Super. Well, I'll wait another couple seconds to see if anybody has further questions. Feel free to unmute as well.
Thanks everyone for coming and listening to me on such a beautiful Friday.
Yeah, thanks for the great talk, Dan. Just a reminder that next week is spring break, so we will not have a seminar, but please join us in 2 weeks to continue on with our seminar series. You can also find this talk recorded at the Conservation Science Seminar website. Thank you very much, Dan, and thanks everyone for coming.
Thanks everyone, bye.